course title is so much related to my specialization. Actually, recent 2 years I was publishing in these kind of an areas, also getting some kind of a grants on this. So I think even if faculties, those who learn, even they can prosper based on, because this is an emerging area in this decade. So I think you have chosen a very nice topic, and you are organizing well. From the beginning of the lecture, I was delighted to find each and every guest lecture so happened very well with both institutes as well as with the industry people. I'm also learning something.
Thank you, sir. Thank you.
Thank you.
So inspiring from you, actually. So just we can wait for 5 minutes, sir, and then we can start. Balaji, sir?
Yeah, good afternoon. I'm here.
Yeah, good afternoon. Okay, sir. Fine. Thank you. Praveen, sir, shall we start? People, in the meantime, people will join. Or can we wait? So you are in mute. You are in mute.
It's your wish, ma'am. It's a recorded session. If they want, they can also.
Already almost 45 participants are there. Still. And probably in the 10 or 5 or 10 minutes, people
okay, ma'am.
Posted message also.
Yes.
Meanwhile, we can complete the site.
So my voice is audible, isn't it? So it's adequate.
Bit louder is better option.
Okay.
Be a bit loud.
Yes, ma'am. I got it.
It's everything perfect. And a very good afternoon to all the delegates joined here for this day 2 afternoon session, which is going to be hosted by Dr. Praveen Kumar sir, and sir is an associate professor in Sri Venkateshwara College of Engineering, Tamil Nadu. And today, sir is going to deliver a talk on AI in drug repurposing for variant strains of SARS-CoV-2. So from the management and principals side, I just welcome you for this artificial intelligence drug repurposing and polypharmacology ATAL-FDP sponsored by AICTE, sir, and the heartiest welcome to you. And very good afternoon.
Thank you, ma'am. Good afternoon to Dr. Sucharita, madam, the convener of Seven Hills College of Pharmacy organizer, and also I also express my good afternoon to all the participants, those who are sparing valuable time to learn, to acquire knowledge, as well as to acquire practical hands-on with a few software techniques. So I think I can share my screen. I can go ahead.
Yes, sir. Yeah, before going for that, just give me 2 minutes of time to honor you with your citation, sir. And I request my dear colleague, Dr. Balaji, sir, to do the need and introduce the stature of Praveen sir to the delegates. Sir, please.
Yeah, thank you. Is my voice is clear, madam? Is I'm
Clear,
audible?
sir. Yeah,
Yeah.
clear, sir.
Thank
Turn on video, sir.
Yeah. Yeah. Good afternoon, today's delegates. Myself, Dr. P.N. Balaji, working as a professor from the Department of Pharmaceutical Chemistry. I take this as a privilege to invite and brief out our today's day 2 speaker, Dr. P.K. Praveen Kumar sir. Sir is working as an associate professor in the Department of Biotechnology and having more than 19 years of teaching experience. Presently, sir is working at Sri Venkateshwara College of Engineering, Chennai, Tamil Nadu. Sir have a core interest in drug design and development, vaccine design and development for virus and cancer diseases. Sir also have worked in bioinformatics tools, including machine learning and artificial intelligence, cheminformatics tools, and wet lab experiments on HPLC, FTIR, ELISA, and FACS, and small animals handling and experiments. Sir has published more than 24 papers on drug and vaccine designs at reputed journals like Elsevier, Bentham Science Publishers, Nature Publishers, Mednock Publishers, and Karachi Publishers, etc. He has also published the three chapters in a Bentham Science Publishers books. He received the Coursera certifications on healthcare and primitive medium genomics technology, drug discovery, and immunology, etc. He is an executive guest editor and reviewer for various international journals, and Sir has also received an Young Scientist Travel Grants from CSIR and ICMR to present his paper in an international conference at Kuala Lumpur, Malaysia. And Sir has conducted various SHCPs, FDPs, and webinars which are funded by the DST, SERB, AICTEs, etc. So by this, I take this privilege to welcome today Dr. P.K. Praveen Kumar sir to give his valuable lectures on artificial intelligence in drug repurposing for variant in SARS-CoV vaccine. Sir, please, sir.
Thank you very much, Dr. Balaji, sir, and for nice introduction, and also feel thankful to Dr. Sucharita for providing me an opportunity to share some information related to artificial intelligence in drug repurposing for variant strains of SARS-CoV-2. I presently shared my PPT. Is that in a presentation? No,
Yes, sir. Yes, sir.
It's in presentation mode. Good.
Yeah, yes, sir. It is in presentation.
So good afternoon to all the participants. Now, I think I can take you to this topic. So highlights of the presentation, artificial intelligence, we basically discuss about the definitions of what is artificial intelligence, repurposing, and how about the artificial intelligence and machine learning approaches for prediction of repurposed drugs for SARS-CoV-2, and how about machine learning techniques and its types, repurposing small molecule databases, macromolecular databases related to SARS-CoV-2, and how about the different strains and computational methods in drug design for COVID-19, artificial strategy for repurposing of drugs for COVID-19, and drug discovery hackathon 2020 team, which I mentored recently, and summary of the entire kind of a presentation. I recapitulate after all the topics. And then we also discuss what are the upcoming events in SVC. So basically, I'm going to teach you I just going to deliver you the information for 1 hour. And the last 20 minutes, we can have a kind of an hands-on kind of an session. And then the last 10 minutes, we can have in the form of a question round. Okay, so I see that I can, but I have up to 80 slides. Okay, now itself I informed you. I see that I can go according to that kind of a speed. If you have any doubts, even you can interrupt me at any time, because even if you want to make more interesting by asking doubts, so you are welcome. Okay, so I think with positive thoughts and positive vibes, I'm proceeding well to the first slide. So about artificial intelligence. So why we have to seek the artificial intelligence? Because we already have natural intelligence. So humans, why we have to depend on machines? The reason behind is to rely on speed and accuracy, isn't it? So only we are thinking about to go for an artificial intelligence. So if you want to use artificial intelligence, you should have a machine learning model. So if you describe the artificial intelligence or machine learning model, it's a software program that has been trained on a set of data to perform specific tasks like recognizing certain patterns. Patterns, I mean here, can be image data or digital data or analog data. Okay, so we have types of data, isn't it? So whatever data you obtain, okay, you have to analyze it. Okay, you have to train in the form of a training data set. Then you have, if whatever you have as a sample, as a test data set, you can use a test data set to determine from the training data set. Okay, so this is a simple thing in artificial intelligence where we use a machine learning models. And if you see the list of the most popular artificial intelligence models, we have linear regression, deep neural networks, logistic regression, decision trees, linear discriminant analysis, naive Bayes, support vector machines, learning vector quantization, and bagging and random forest algorithm. Drug repurposing. What is drug repurposing? Till now, we have seen what is artificial intelligence and the models. Now, we can also see what is drug repurposing. It's also known as drug repositioning, which is a process of identifying new therapeutic use for old existing available drugs. So we are seeing past lectures from industrial persons like Dr. Girinath Pillai, Dr. Divya was explaining about more about the drug purposing as well as machine learning. So I just gave some kind of definitions, and I think I can proceed well. So why we have to go think about to go for drug repurposing and artificial intelligence? Because if you see the GDP, okay, the GDP in India also lies on how the pharma industries grows, okay. And if you see in overall and globally, pharmaceutical industries have significantly played the market for repurposed drugs alone at $24.4 billion in 2015, with a projected growth up to $31.3 billion, not rupees, $31.3 billion in 2020. You know, most of the drugs, okay, whatever we use for treating various diseases, we use up to 30% of repurposing drugs in daily use. And if we see the artificial intelligence and machine learning, whether this can be read out, I mean, I don't know, something is blocking at the top. Actually, this is an artificial intelligence and machine learning approaches for screening drugs for various coronavirus diseases. If you see SARS-CoV-2, okay, which came as pandemic, okay, in March 2019, isn't it? So in February, I mean, in December 2018 itself, we can find its origin in Wuhan, okay, where they identified that a virus as SARS-CoV-2. But you know, this is not just a new virus, new kind of a coronavirus is coming from China. Already we have got some SARS-CoV-1. Okay, so in 10 years before, in 2003, and even in 2007, we have SARS-CoV-1. In MERS 2003, it seems. Okay, so Middle East respiratory syndrome. If you see all these things comes under coronavirus family, where if you see these kind of entries they have taken from Akanksha, who published in Computational Structural Biology. According to her, she has taken this much kind of an entries, and she she has developed a kind of a Python programming algorithm where she has used a package called sklearn. Okay, she used a Python programming, and through the Python programming, she used machine learning packages like support vector machine, random forest, artificial neural networks, nearest neighbor, deep neural networks, etc. These machine learning techniques she has used, and she optimized the predictive models. Later, she has discovered that drugs, repurposing drugs, whatever we use in SARS-CoV-1, they tried with SARS-CoV-2. Once a successful happened is remdesivir. It's a popular drug. Generally, it targets RNA polymerase. Okay, so the same kind of a drug, okay, of course, is also approved from FDA for treating SARS-CoV-2. Okay, it's a major drug used all over the world. And the later we go for the molecular docking. Okay, molecular docking and simulation studies. So she carried out at last to validate the result. So this slide explains you about the different machine learning techniques. Initially, I've taken support vector machine. Okay, in this support vector machine, we have two types of data. One is red dots and one is black dots. Okay, suppose if you think like the red dots or the molecular weight is above 400, and if you have black dots like less than 400, if you want to distinguish the drug candidates, okay, based on the molecular weight, you can draw your hyperplane. So already you have two hyperplanes. Even I can draw the hyperplane like in such a way that I can able to categorize, I can classify major kind of the drug candidates. Okay, so we can have a kind of an hyperplanes. If you create a kind of a good kind of an hyperplane where you get a more positives, more instances, okay, then you can try to use this kind of support vector machine algorithm. So this is actually called as data classification algorithm. Okay, generally used for data normalization problems. One such example already we have find is a Swiss ADME. Okay, this open source software. Free software can accessible in internet. You can able to find in this image is a boiled egg. Okay, so this is an image of boiled egg. I think many faculties have experienced using the Swiss ADME. I'm just linking with how the support vector machine integrates with the this kind of software development. So in this boiled egg, okay, platform, you can able to see the yellow color. It looks like an egg, isn't it? So when we call this a boiled egg diagram, where this is actually blood-brain barrier. So why we have a blood-brain barrier as a yolk of an egg? Because it's more. What is the reason? Can I get a reason why we have a blood-brain barrier inside present as a egg yolk? I think if participants know, even you can interact. So that we can make it's interesting. Very good. Exact answer is lipophilic. So congrats to Arunji, he has answered well. So you have more lipophilic in the egg yolk, okay, when compared to the egg protein, albumin protein. So if you see here, our drug is lying somewhere here, isn't it? So our drug is lying. So yes, is lying here, which is not a lipophilic, but it's a more hydrophilic. Okay, and if you see this kind of categorization of data, okay, can be happen with SVM, like how I explained. We had a kind of a hyperplane for like a yolk sac, and we distinguish that. And similarly, here you can come across the utilization of the next machine learning technique, deep neural networks. In deep neural networks, we can use the best example is deep screening. What is first of all deep learning neural networks? These are actually used for analyzing. We use generally terms like input layer. We have a different kind of an inputs, and we can have as many number of hidden layers as we want, and we can also have a kind of an output layer to generate the result. This is something like we have an activation functions. Okay, we're using the activation functions if you want to screen the drugs. For example, if a person is a handicapped person, okay, so think like a person is leg handicapped. So generally, if he is like a handicapped, he may come across with some kind of side effects, like he may come across with more leg pain. He may come up with a kind of a blood pressure. Okay, he may come up with some kind of a respiratory disease like an asthma. He may experience. Okay, we may expect. Okay, so if he is diagnosing in the hospital, and if we based on the intensity of pain and based on the drugs, whatever they subscribe for a particular disease. For example, if he is going to treat for a fever, and whatever drug he is going to take for a fever, it should not give a side effects with any of these kind of. And so this is what actually generally we call it as a morbidity of drugs. So if you want to check one drug may leads a kind of a side effects with another kind of an illness, isn't it? So we have to check even how the drugs act, behave like a synergistic action, or it gives a kind of drug regulation, okay, so gene regulation, so all these things matter. So we have as many number of hidden layers as we want. And finally, if you want to get a kind of a drug for the disease, what exactly we want to treat, okay, based on the kind of an illness, we can give a kind of a drug, even the dosage form can be fixed like that, okay. So this is a kind of an application of a neural networks, of course. And then example they have provided is a deep screening tool, okay, where we can able to use a neural networks for screening the drugs. And also this deep learning technology can be used for analyzing medical images, for image acquisition, analysis, all this. We can also use deep learning neural networks. The next machine learning technique, what we are going to see is the k nearest neighbors, where the best example of the software, what you're going to see here is Zinc 15 docking branch. Okay, if you see this software, okay, then later I can explain you what exactly is k nearest neighbors. So here in this example, okay, we have a two-dimensional data in that table, molecular weight and as well as the log partition coefficient. If you see a small molecular weight compounds, okay, so generally we take nearly 5 important properties for estimation of drug likeness properties. And if you take any of the 2 properties from the drug likeness property, and if you plot in the form of a table, this is already available in Zinc for 15 docking open source software, okay, where you can able to see, for example, if our compound molecular weight is lying in 425 and partition coefficient is 3.5, then exactly our compound is one of the compound from 13 million molecules. Okay, so this is how we can able to check. Okay, so nearly 1.3 billion molecules are there deposited in this Zinc 15 branch. And so how we identified, okay, our molecule with a neighboring molecule existence, like even if you want to check for any kind of a docking or any kind of a the next advanced simulation studies, okay, we are checking with the more closely resembling the parameter attribute compounds with respect to that kind of drug design studies. You got it? So if you see this k nearest neighbors, okay, it's a machine learning model which works extremely fast and more accurate and efficient at finding the needed values of test data set. So this is, of course, a test data set, not entirely the 1.3 billion compounds we take in existence. Presently, for we have nearly 997 million substances are deposited, okay. But I think also few more molecules, each and every day they are depositing few molecules in the Zinc 15. Okay, so it may we may experience up to 1.3 billion soon. And if you see the prediction of the outcome value are calculated by checking the whole data set for k data nodes with similar values, so-called neighbors. So if you see our drug candidate is lying inside this circle, okay, which is closely resembling with that blue kind of candidates. We have 2 types of candidates, isn't it? Blue and orange. Suppose the blue is more hydrophilic and orange is lipophilic. We can, based on the nearest neighbors, we can able to categorize it as the hydrophilic, okay, because we have 2 blue when compared to only 1 orange, okay. So this is how even we can able to estimate the values from the k nearest neighbors. And we can see in another machine learning technique called random decision forest algorithm, where you can see an application, already an application is developed using this algorithm, that is Galaxy QSR. Most of the QSR softwares are developed using the random decision forest, where here in this example you can find 3 different trees, and each and every tree decision is different, okay. So based on the inputs, and if you want to get what kind of an output, okay, so we can select the kind of a tree. So random forest trees are formed of decision trees where multiple samples of data, okay, so each and everything is a sample now or processed by decision trees, and the results are aggregated to find more accurate output value. And of course, this gives you a nice decision to choose even drug, okay. And if you come across the next machine learning technique is just in neural networks. In neural networks, we have types. If you see in neural networks, as I already mentioned, we can use for image analysis, isn't it? So if you see a SARS-CoV-2 patient, okay, generally we take image data from X-rays. Okay, so what exactly we do? We take an X-ray of lungs, isn't it? So based on the black and white convolutions, so the spots, okay, present in the lungs, we can able to find how much SARS-CoV-2 infection has occurred. Okay, if you find like this, like here you can see, so you find you don't find much black portion. So you can find much SARS-CoV-2 infected, okay, when compared to the other images. So likewise, okay, so but if you by detecting through natural intelligence is somewhat difficult. So we pass it to heat map, okay. So we use one method called heat map, where based on the color, okay, the red and yellow, we can able to identify how much SARS-CoV-2 infection has occurred. Okay, so this is developed in dark COVID net kind of a platform. Okay, and if you want to use this kind of a machine learning, okay, we can use softwares like KNIME, as just now Dr. Girinath Pillai has explained how to use the KNIME, how to construct a kind of pipelines to integrate and to determine the chemical compounds, so chemical descriptors, all these things he has explained. So this is an example how you interlink each component in a neural network and how to get an output from the inputs what you feed. Okay. Similarly, if you are using a large data linked with cloud computing, we can go for TensorFlow. I think recently I guided a PG student with the TensorFlow application. Actually, this we link with Python. Okay, so for Python linked with cloud computing, we can go for this kind of TensorFlow and Vika on RapidMiner. Okay, so most of the softwares or commercial softwares, okay, to handle the image kind of a data. And in this image, we can able to see the image in silico analysis of drug repurposing. Generally, if you go for the drug repurposing, we can either go for in silico approaches or experimental approaches. In experimental approaches, we generally go with in vitro or in vivo experiments with animal models. So if you go for in silico approaches, where our major FDP program focused on, okay, we can either go for drug-based, okay, or we can go for disease-based or target-based. If you go for drug-based, okay, we can check for the similarity of the drugs, okay, with the existing drugs, okay. So that is how the drug repurposing is done. And we can also go for a kind of a pharmacophore similarity. We can check for metabolomic kind of a data, and we can go for chemo informatics and the side effects and adverse effects of the specific drugs. And similarly, if you go for target based, so we generally target protein. So protein interaction networks, and how about the gene expression data and networks, and how about the interaction of our compounds with respect to the different genes or related to the proteins. So all these things we check with molecular docking. Later, the stability of binding we check with the molecular dynamic simulation. Okay, and the disease space is, of course, is linked with the drug base. However, whatever we see, we also see here. Other than that, we also focus on specifically epigenetic data and phenotypic kind of a data. And if you go for the strategies of drug repositioning with examples, okay, here you can able to see we start with a drug, okay, whatever repositioning drug we discover, we try to discover with various targets. And each and every target is linked with different pathways, and from the pathways we can see it's what disease is it associated and how about the side effects for a drug. Okay, so this is a general methodology, and if you see in the form of strategy, the strategy 1 to strategy 7, we can able to observe according to Mithun et al. So he discovered the first strategy as a molecular docking and pharmacophore modeling approach, where he used virtual high throughput screening, or we can also able to screen through the high throughput screening or high content screening, okay, in terms of in vitro studies. Okay, so according to him, he determined the reposition drugs for SARS-CoV-2 as bupropion and thalidomide. And similarly, strategy 2 is related to chemical similarity or fragment-based screening. That is a kind of a genomic approach, and based on a proteome approach, we can able to study through biochemical assays, cellular assays, animal models, and these are the drugs that he has come across. And for based on transcriptomics approach, that is based on protein expression, gene expression, these drugs are the result of this repositioning technique. And through metabolomics, the dual-excitin, which where we need a signaling pathway or omics data mining, and through bioinformatics generally or chemoinformatics, aspirin and metformin. So this is actually a biomarker-based assay, and so this is not exactly SARS-CoV-2. He actually focused on drug repositioning kind of a studies, and for a different disease, he come across types of drugs. Okay. And we in this slide we can see the yes, I think now only. Okay, so in this slide we can able to see the literatures from NCBI PubMed. Okay, I think this snapshot I've taken 3 days before. Nearly 1.63 lakh publications are done with COVID-19. Actually, the COVID-19 raised in 2019, and the past 2 years itself, we found nearly 1.63 lakh kind of literatures. But if you see the non-infectious disease, but most dreadful disease in India, that is caused because of malaria. This malarial viral disease from 1828 to 2022. So obviously, it's more than it's up to 200 years, isn't it? So up to 200 years, we can find only up to 1.02 lakh kind of literatures or published. And if you see the COVID-19, the percentage of fatality is more because of infectious, and because of non-infectious, the percentage of fatality is less. But if you see, okay, for 2 years, we find number of cases 214.5 million, okay, all over the world. But in a single year, the last year itself, okay, the number of cases affected by malaria is 229 million. So obviously it's a 229 into 2. Okay, so we have more patients affected by this kind of viral, but because of non-infectious, the personal fatality is less. So this makes us to focus more on this kind of infectious SARS-CoV-2. And if you see the computational literatures, we find up to 11.75
Praveen sir? Hello, sir. Hello, sir. Praveen sir, can you hear me? Yeah, delegates, I think there is a small technical glitch. We'll get back to you in a minute. Yes.
So I'm audible. Kindly excuse, I have some internet connection streaming problems. I think I shared my slide.
Yes, sir. Yes, sir. Yes, sir.
Thank you.
You're audible also.
Yeah,
Continue,
thank you. Protein interaction information we can obtain from these databases. So I think I can move to the next slide. So information available from pathways related we can find in NSA pathway interaction, cake and pathway commons, and clinical trial information we can obtain in clinicaltrial.gov. Okay, even in NCBI we have a clinical trial database, and in omics we can obtain from NCBI gene expression omnibus, sequence read archive, and cancer cell line encyclopedia. So the corresponding URLs of the websites are mentioned here. And in this slide, we can going to see the repurposing small molecule databases. The first is NCBI PubChem. So up to 2 days before I updated, so nearly we find 1,605 compounds related to COVID-19. And this, so most of the compounds are plant compounds, and we have some substances, 65 substances, 654 genes, 546 proteins are associated with SARS-CoV-2, where it can be linked with 2,278 pathways. Then think about the complexity. If you go for transcriptomic profiling, okay, so obviously you have to link with all this kind of a pathways to find the exact kind of a drug. So when we can't able to have a kind of a an exact kind of a drug for COVID-19, we are just using the repurposing drug. Okay, so the whatever FDA has approved is going to partially treat the patients. And bioassays, we find 282 literatures, we find up to this much. And ChemSpider database. So this is one more database where we find a repository of organic molecules as well as the synthetic molecules and drugs, etc. And CHEMBL, you can find similarly the chemical compounds and drugs, etc. CHEBI, this is one more database. And drug bank, majorly, this is a fully annotated database where you can find pharmacology, interactions, products, categories. Like if you want to purchase any kind of drugs or compounds, whatever deposited in drug bank, you can also purchase, you can have the vendor's information, and you can put a quote and you can purchase it. Okay, so almost you not only limited up to that, we have some kind of targets, enzymes, transporters. So almost all the informatics related information you can find in this drug bank. Zinc 15 database. So we have nearly 230 million purchasable compounds ready to draw, but also we have 750 million purchasable compounds. Okay, that you can search for analogs in under a minute. This is what actually in 2019, when on March, it was discovered as pandemic. Okay, COVID-19. When COVID-19 was declared as a pandemic, within 2 months from California Institute, a scientist has done a kind of a virtual screening with this 1.3 billion compounds, okay, in a supercomputer, and he discovered with few compounds. So one such a few compounds, what he discovered is remdesivir and a few plant-related compounds. Accelera COVID-19 drug repurposing database. This is an exclusive kind of a COVID-19 drug repurposing database where we find few thousands of molecules deposited. So where you can also find the target information and other kind of literature supported information in this database. And next database, what we're going to see is binding DB database. Okay, where we can find even the drug dosage levels, pH, and the interactions, all these things we can find in this binding database. AICTE database, so this we call it as a Kyoto Encyclopedia of Genes and Genomes. This is a database hosted by Japan. You can find the pathway related drug, enzyme, etc. These are the major data entities we can find in this cake. Okay, so major pathways we can find in this database. Stitch pathway database, where you can find how a drug is regulating each and every proteins, okay, and how it's associated with other kind of drug like morbidity and synergistic kind of an relation we can study with this kind of a stitch pathway database. Small molecule pathway database, where in this database you find the small molecules, like you can search for a small molecule related the interaction, like a molecule, the small compound interacting with the protein. So the whatever information. Okay, so I'll be showing you through the hands-on. Okay, so this is just a small molecule pathway database. When during the hands-on, if you have any doubts, you can ask me. Okay, so this is something related to interaction patterns we can discover in this database. And now we see the macromolecule databases, because if you go for a kind of a drug repurposing, we need a ligand as well as a receptor. Till now, we have seen the ligand repository databases. Now I'm going to show you the macromolecule databases. So of course, immediately we come across the RCSP PDB, Research Center for Structural Biology Protein Data Bank is the largest repository of 3D structural protein database, where we can find around 1.282 lakh structures are deposited by gold standard techniques like X-ray, NMR, or cryo-electron microscopy techniques. And the next slide, we discuss about MMDB, molecular modeling database. So we find next to the PDB, we find more protein structures in this. So we can find 1,558 molecules. But earlier, if you see in NCBI, RCSP, we find nearly 1,460 proteins are there for SARS-CoV-2. Okay, this was updated yesterday. But here you find somewhat more molecules when compared to RCSP PDB. Okay, because NCBI is maintained by National Institute of Health USA. So they have taken extensive efforts to deposit more proteins, majorly through cryo-electron microscopy techniques. So you find more structures inside NCBI MMDB when compared to PDB. And if you see the computation methods in drug design for SARS-CoV-2, I've listed out around 10 different methods. The first method is the drug likeness prediction. We can do with the MolSoft mole inspiration. These are free online softwares, and ATMET prediction can be done through Swiss ADME pre-ATMET. So all our online softwares, whatever I listed in black and whatever listed in blue, or the commercial softwares. DS, I mean Discovery Studio. So for quantitative structure activity relationship and quantitative structure property relationships, we can use these softwares. And pharmacophore modeling, we can use PharmaGIS, Mapper. So we have plenty of methods, isn't it? Here, whatever methods we see here, okay, these are coming under ligand-based screening. So this comes under ligand-based screening. Okay, so this is a hybrid method. Okay, and if you want to go for a receptor-based screening, of course, we need molecular modeling, docking, inverse docking, dynamics, all these things. Okay, this comes under receptor-based screening methods. Okay, this comes under hybrid, and of course, read across approach. We need it's a kind of a ligand-based approach. Okay, so the tools, whatever I shown, can be predicted according to the OECD guidelines. That's what Dr. Divya Madam explained, you remember? For toxicity prediction, we have to follow the global OECD guidelines. So the softwares are developed based on that. And this is, of course, a free software, Cytoscape, used for determining the gene-gene interaction networks, protein-protein interaction networks. This is a freeware. Now, Autodoc we use for docking ligand with respect to receptor, and we can also use machine learning techniques developed through the Python R programming. Okay, am I audible?
Yes, sir, you're audible.
Any doubts up to present? Like participants, if you want to ask anything, you can ask me, or I can continue. I think I'm explaining only the basic techniques, basic methods, but still those are new to the bioinformatics. If you have any doubts, I can raise.
Yeah, you can continue, sir. I think there is no queries in the chat box.
Thank you, ma'am. So drug likeness prediction. So this is the first method of drug design where we use many filters. One such a popular filter of drug likeness is Lipinski rule, where we try to filter this filter is generally used to determine whether your plant compound is acting like a drug or not. Generally, we use it for kind of a plant compounds. Okay, like if a plant compound proved to be a drug, okay, it has to satisfy any 4 properties. Okay, even any 3 properties we can consider out of 4 properties. First property is the molecular weight. The molecular weight should be less than 500 gram per mole. And the second property is from the partition coefficient, that should be less than 5. The partition coefficient, whatever we are going to calculate, is octanol water partition coefficient. That should be less than 5. Okay, and hydrogen bond acceptor should be less than 10, and hydrogen bond donor should be less than 5. If any plant compound, okay, satisfies any 3 out of 4 properties, still we can tell that our plant compound is acting like a drug. Okay, now in addition to these 4 properties, we also have rotational bonds, which should be the acceptable is less than 10, and polar surface area should be less than 140 angstrom. Okay, and to check the drug likeness properties, that is especially using the Lipinski rule, also called as Pfizer rule, we can use softwares like MolSoft, Mol Inspiration Freewares, and also we can use Swiss ADM. You know, even in NCBI PubChem, that is a database, we generally repository of chemical molecules in NCBI. Okay, this also you have a repository of data of this five the Lipinski rule of 5 parameters. Okay, why we call it as a rule of 5? Because all these 4 properties are in multiples of 5. That's the reason. And if you see the output of drug likeness, okay, we can able to find for a particular compound. Okay, the score is mentioned as minus 0.82. So it means that okay, if a compound act to be a drug, okay, the range, acceptable range of drug likeness should be minus 1.5 to plus 1.5. So even from this kind of through the correlation, they have developed a drug likeness model score. This kind of a result we obtained from in MolSoft, our drug is lying in this region, so we can tell our compound, this compound is acting like a drug. Also, okay, for this compound, the hydrogen bond acceptor is less than 10, and it is less than 5, and it's less than 500 gram per mole. Okay, so 3 properties already satisfied, and the 4th property is partition coefficient. This also is less than 5. So almost all the 4 properties satisfied. So very well, we can accept this as a drug. Okay, this is how we can determine the drug likeness properties for a plant compound. And the next is to check the pharmacokinetic properties through ADMET descriptor calculation. Actually, what is ADMET? A for absorption, T for distribution, M for metabolism, E for excretion, T for toxicity. So this is what we call it as a pharmacokinetic properties. So in addition to that, we also have a liberation, like from buccal activity, what's happening, okay? So how the drug is releasing. So this accounts for a liberation. So absorption, how a drug is going to absorb, okay, in the blood, okay? This the absorption of drug in the blood, we determine using this absorption parameter. Also, absorption can also, after absorption, we come across the distribution, isn't it? So in distribution, okay, from the blood to the tissues, like for example, if a patient is suffering from lung disease, he have to target only the lung. He don't want to target something else. So if you want to target the lungs, so the distribution should happen only in the lungs, not somewhere in kidney or somewhere in some lymph nodes. Okay, if that has happened, that has to be happened. Then the drug should have a the kind of adequate distribution properties to the lung. Okay, and if you see the next property is metabolism, we know that the first pass metabolism happens in the liver. Okay, more than 50% of the drug, okay, is utilized in the liver. So bioavailability is will be reduced. Okay, if much of the compound dissolves in the is metabolized in the liver, so we should also take care of this property. And excretion is the final, like excess drug is excreted in urine. Okay, so this 5 properties. Okay, I mean 4 properties and 2 minor properties are also determined through Swiss ADME, as I already explained. Also, we have some few commercial softwares to determine the ADMET characteristics. And one such output derived from Swiss ADME is mentioned here. So here you can find the Lipinski rule. Okay, so you can find molecular weight, you can find hydrogen bond acceptors, donors, partition. Other than the partition coefficient, we also have total bonds. Okay, total polar surface area, as I mentioned. Okay, so we also account for this. The absorption we can find, like if your gastrointestinal absorption is high, so from that you can determine how effectively your drug is going to absorb in the blood, and how about the blood-brain barrier permeate. Like if whatever drug you take, it should not be, it should not affect CNS, okay, the central nervous system. Then you should take care of whether your drug is a lipophilic or hydrophilic. Okay, so if you have more lipophilic, may your drug also penetrate to the blood. Okay, so we should take care that our drug should not penetrate into the blood. So we have to check our absorbed properties. No. Okay, if it is yes, obviously it can be used as a CNS drug. Okay, not a normal drug. And we also have the cytochrome P450 enzyme related kind of an activities. Okay, so this is related to distribution. Okay, and of course, based on the dosage, okay, based on the solubility, even we can able to find how much of drug is excessively excreted, okay, in the form of urine or sweat, etc. Okay, so this is generally the adiabatic properties can be checked or using this freeware Swiss ADME tool. And pre-ADMET is similarly like that. Okay, in addition to drug likeness prediction and ADME prediction, we also find toxicity prediction. Okay, so through rodent carcinogenicity assay and IMS test, so all these things are already models are developed. So all these models are developed through the languages, of course, through artificial intelligence and machine learning techniques. QSAR, quantitative structure activity relationship, this is a ligand-based screening. Okay, I think Dr. Girinath Pillai has explained in detail. Okay, so simply I'm giving you the basic definition. We generally how to predict it. We have to initially predict a model. Okay, we can also call it as a training data set. And from the training data set, whatever ligand we have, whatever drug we have, if you want to predict the activity of our drug with the existing drugs, we can use this kind of a QSAR. Okay, for example, if you have 10 drugs and 10 different drugs activities are known, you try to develop a model for the 10 different drugs. Later, whatever drug you have, a sample one drug you have, you test with it, okay, using this QSAR studies. From QSAR, like QSAR, we have, if you study property instead of activity, if you focus on property, we call it as a QSPR. And if you focus on reactivity, also, I think in a receptor, you come across interactome, isn't it? So the similar, the interactome related kind of studies, if you study with respect to small molecule compounds, we call that as a reactivity. Okay, how a drug is reacting with another drug. Okay, whether we are getting a kind of a morbidity or synergistic or so what kind of drug inhibition. So all these things we study in QS RRs and QS chromatography relationship. Some plant compounds are very close related. For example, if you take ethanolides, okay, we have 35 different types of ethanoid compounds, okay, in a plant. And if you take steroids, some group of steroids will be there in a plant. So, but slight distinguish will be there. Okay, molecular weight, of course, may remain same or may have a 1 or 2 plus or minus 5 kind of a gram per mole. Okay, so all these things can be clearly distinguished in this chromatography relationships and toxicity relationships. We can determine through QSTR and electrochemistry relationships through QSER, etc. And reverse talking studies. This is actually we call this as a target deconvolution. Okay, generally we have a kind of a drug, and from a drug we dock with a kind of a target receptor and find whether our drug is effectively binding or not. If our drug is not binding, we check with another drug, isn't it? So we have a series of compounds or drugs to be checked with a single target. But here in reverse talking, it's quite opposite. We have plenty of receptors, a group of receptors are provided, and you can choose only one compound. Okay, if through one compound, we can find whether our compound is talking effectively with what kind of a receptor. Okay, with a single compound, we check it's binding receptors. Okay, which is binding well. For example, for this plant compound, I think this is binding effectively with oxidoreductase. So this is a kind of an oxidoreductase inhibitor. Okay, likewise we can determine. Okay, so this is what we call it as a target deconvolution studies. Okay, this can also we call it as reverse talking. So this can be performed using acid that is auto in silico constants inverse talking studies. And pharmacophore mapping. Okay, so of course it's a category of a pharmacophore modeling. This can be performed using zinc farmer, bicalin. Okay, actually, what is a pharmacophore? It is an abstract description of molecular features that are necessary for molecular recognition of a ligand by a biological macromolecule. Okay, so we generally try to discover a kind of a pharmacophore. We'll go for a pharmacophore mapping or a modeling to find the steric and electronic features. These are the major parameters they focus. Okay, so like if you see here, okay, mostly the hydroxyl groups are more prone. So what are hydroxyl groups? Hydroxyl groups are coming under polar groups, isn't it? So these polar groups generally attract with kind of a non-polar groups. So H generally interacts with the non-polar groups. Okay. So likewise, okay. So for if you see the polar non-polar kind of interactions, okay, obviously we need this steric and electronic features. So pharmacophore mapping or modeling. is based on that. And the next is the QSR, as I already explained. I just showing you a kind of a snapshot of Galaxy QSR. This is a free software tool where we can build the QSR model, and we can later building the model, we can predict activity using different QSR model studies. I think they have utilized two different QSR models. Okay, so you can use any of the model, like from a training data set. Okay, the test data set can be identified. Okay, so as Dr. Girinath Pillai has already explained you, okay, so it gives you more kind of an accurate results because of R square. Okay, this gives you the precision kind of a results. Okay, and about SARS-CoV-2 and its strains. If you see the SARS-CoV-2, it is a 29.9 kind of KB. Okay, the genome size of the SARS-CoV-2 is nearly 29.9 kilobase pairs, where you can find majorly 20 proteins, four structural proteins or spike, envelope, membrane, and nucleocapsid. And we also have four non-structural proteins, so that is NSP1 to NSP16. So RNA-dependent RNA polymerase, one of the chief target also lies in the one of the non-structural protein. Okay, and if you see the most important target protein in SARS-CoV-2 is nothing but spike. Okay, because this spike glycoprotein plays an important role in connecting with the ACE2 protein of human host. So just because of the host, we find the viral entry, and so we majorly concerning on to target this spike glycoprotein. If you check for the kind of results, okay, that is a number of proteins deposited in PDB, as I already mentioned, we have nearly 1,423 in PDB, RCSP PDB database. And if you see here in this slide, we can find our spike glycoprotein, which is the chief, the most important target, binds with the ACE2 of human. Okay, so generally it binds with the receptor binding motif domain. Okay, so the red color, whatever I shown here, is the receptor binding motif, so which is interacting with the ACE2. Here, even if you want to discover a kind of a drug, okay, which blocks the kind of a receptor binding domain, we have to face some kind of a challenges. And if you see the receptor binding motif, okay, so if you see the receptor binding motif, okay, it lies in around 437 to 508. Okay, so around nearly 68 amino acids are important, and out of 68 amino acids, we can find 13 important amino acids, or so these 13 amino acids, these 13 important amino acids are very important, which is connecting with the human host. Okay, so if your drug targets with any of these 13, you may get a kind of more efficacy with the kind of a drug. Okay, so the efficacy can be increased if you have more contacting binding sites. And if you see in this slide, okay, we have types of states of spike glycoprotein. The important challenge in discovery of drug with respect to spike glycoprotein is like here we find the spike glycoprotein in both closed state as well as in open state. Okay, if you see in open state, okay, much of our the spike glycoprotein is interacting with the ACE2 receptor. But in closed, you don't have that much kind of an interaction. Okay, so in open, you have a kind of interaction. Okay, whether you can see the open here, you can have a good kind of interaction, but here you don't have that much kind of a good interaction. So if you want to interact effectively well, okay, that should be in semi-open state. Okay, when compared to open and closed, if your drug is targeting the spike glycoprotein in semi-open state, you can better still increase the efficacy. Okay, not only because if you go with a closed state, obviously you may not end up with binding with the specific binding, that 13 binding sites of the amino acids. If you want to bind with the 13 binding site amino acids, even the position of spike glycoprotein, okay, is important. So that's what the Mert et al. Has mentioned, that transition to the semi-open state is a critical step in post-cell recognition of SARS-CoV-2. So based on this, even the drugs can be developed. And if you see the spike mutations of SARS-CoV-2, Okay, we have nearly 8 different strains up to present. Okay, this information fetched from CDC. Okay, also we can find this information wiki. Okay, nearly we have 4 important strains, alpha, beta, no, totally 8. We have shown you the first 4 strains, alpha, beta, gamma, delta. 2 strains we find origin in India. Okay, the delta, we find one such strain or originated in India in October 2020. And similarly, we have the next strain kappa. Okay, so the other 4 strains are eta, lota, kappa, and lambda. So totally, we have 8 different strains of SARS-CoV-2 up to present. And if you see the mutations in spike glycoprotein, okay, we have nearly 4 kind of mutations, single point mutations, okay, or 2 point mutations. We find only 1. Okay, I mean, we have 2 2-point mutations, and we have 2 single point mutation, okay, in alpha variant. Similarly, we can find some kind of mutations, okay, with respect to the other kind of variants in SARS-CoV-2 variant. And if you see in eta variant, we observe in 23 countries, and theta in 18, lota in 18, and kappa is in 54. Okay, whatever you're going to experience or whatever you're speaking related to the second wave is something related to the delta variant. Okay, so this is what we are majorly concerning on. Okay, so majorly infecting the children. And if you see the molecular modeling studies, okay, this is another kind of drug design studies where if you want to model a protein based on the spike glycoprotein mutation, for example, you can take it, okay, you download a spike glycoprotein and mutate a particular amino acid. For example, the lysine of 417, if you mutate to 309, obviously it is considered as an alpha variant. So later you can go for modeling. Okay, so take a sequence, you change a character and go for a modeling. After developing a 3D structure, you can go for the drug design kind of a studies. Okay, so this kind of a modeling we can do with any of the molecular modeling studies. Number 1 is the homology modeling. Number 2 is the ab initio. Also, we have a third method like threading. To perform homology modeling, that is taking a target sequence and a template PDB to determine a 3D structure of an unknown protein, we can go for homology modeling method. But an ab initio is a stepwise method. We build from primary structure to the quaternary structure in stepwise procedure. For this, you can use Rosetta, ITASR, Pepford, Robetta. Of course, Swiss modeler we use majorly for homology modeling. And if you see the molecular modeling validation, that is after doing homology modeling, ab initio, or even the threading, we can go for checking the stereochemical quality. If you want to check the stereochemical quality of a protein, okay, that is through 5 psi kind of an bonds, okay, interaction. So we can use the Ramachandran plot. Okay, you can use a Ramachandran plot. So generally we expect a minimum 80% favorable residues. Okay, in Ramachandran plot. If you get a stereochemical quality, that is fair number of favorable residues 80 and above, we can use that protein well for the next advancing studies, advanced drug design studies like molecular docking, molecular simulation, etc. Okay, so we have to check the quality of a protein before proceeding it to for the binding or stability studies. And molecular docking is nothing but the interaction of a ligand and receptor. We speak with respect to energy values. Okay, we speak with respect to dot score or energy values. So different softwares uses different search algorithms and different scoring functions. Okay, so based on the algorithms and scoring functions, we can able to determine the binding affinity of a ligand with a receptor. Okay, so here article I just talked a Nipah virus epitope. Okay, so the Nipah virus epitope with MHC2. Okay, this published in CCHDS. I think this project got funded from Tamil Nadu government and also from intramural, and also we got selected for INEA award in the finalist. Okay, but students luckily didn't go to the New Delhi for presentation. So of course this is considered as one of the best project, okay, up to national level. So where we used autodock for docking a peptide with the MHC2 molecule. Okay, so this freeware you can use. So today I'll be explaining you the demo with the autodock Pyrex. Okay, there's a virtual screening software. So how to do the virtual screening using the Autodoc Parex. And molecular dynamics, because we don't have much time, isn't it? So I can tell you only one technique at least today. So molecular dynamics. So what is molecular dynamics? If you have already a docking to check the binding affinity, molecular dynamics is generally used to check the stability of the binding. Okay, so to analyze the physical movements of atoms and molecules. Okay, so if you see the previous see here, we have a bond stretch and bond torsion, and we have bond angle bend. So this we calculate as bonded interactions, and non-bonded interactions we generally calculate from van der Waals and electrostatic, also called as weak forces of attraction. So whatever energies we calculate from the Newtonian equations of motion. We call this as a classical mechanics. We use classical mechanics, and from the number of molecules we go for statistics. We calculate the sum of forces using the statistics. We call that as statistical mechanics. And if you try to study in the form of wave patterns, okay, we are subjecting to quantum mechanics. So at most, whatever we do in vitro or in vitro experiments to determine the molecular weight of a protein or other parameters of a protein, all these things we are checking here in simulation. Got it? So the quantum mechanics we are using in molecular dynamics simulation to check the stability of a molecule that is a drug binding with the receptor. Got it? So for this, we use softwares like Romax, Amber. So this is a freeware Romax. Okay, will run with the Linux operating system. We also have as a Discovery Studio kind of a simulation software. And one such output analysis. So this is actually output got from Nipah virus epitope binding with the MHC2. Okay, RMS. So RMSD versus time. Okay, RMSD we call it as a root mean square deviation. So according to this, okay, I have run the simulation time for a period of 200 nanoseconds. That is, this complex is the input I have taken, and I have done the simulation using the Promax. So at this time, so almost nearly 150 seconds, okay, 150 nanoseconds, I can able to find a kind of a stability. But the stability originated here, okay, nearly at 25 nanoseconds, we find a stability. Okay, we find a straight line. So later you can find a variation with respect to stability after 150. So this is how we can infer the RMSD output. Okay, that is root mean square deviation, and root mean square fluctuation based on the protein flexible residues. Okay, here you can find at nearly 195 to 120 something. We can find a kind of a flexible kind of a residues observed okay, in this region, and area and the radius of variation gives you the flexibility of a protein. And if you see in this, this is the next method of drug design, that is expression profiling. We call this as transcriptomic data profiling. A recent data published in the Informatics in Medicine Unlocked. Okay, so where I have used a kind of a gene regulation. Okay, so I found out how much positively regulated and how much negatively regulated, because a drug discovery for a disease don't rely just up to the upregulation. May the downregulation of a plethora of cytokines or the immune related proteins may influence change the effects with respect to a disease. Okay, it may give an adverse effect or it may give an effect. So all these things we have to counter. Okay, so we have to take all those proteins, immune-related proteins related to a specific. I have taken a case study as a ovarian cancer. Same you can even take for any viral disease. You can take a viral kind of a receptor spike like a protein, and I can link whatever proteins related to spike like a protein, and then after finding the positive and negative regulation, the top regulated genes based on the score, you take some 10 important genes. Here I have taken for the 10 important genes related to ovarian cancer, and I checked with binding studies with the 3 important plant compounds. I found quercetin is found to be better with good values when compared to the other 2 compounds. So similarly, we can do with SARS-CoV-2. We can do with spike glycoprotein linked with other proteins. The screening we can also do with a zinc database, as I already explained. Okay, it uses kind of a neural kind of a what is this? So this uses a kind of a machine learning technique to work drug design. And CHEMBL, where we can discover the drug from 14.6 million bioactivities. Okay, so already we have literatures Okay, done with all the 14.6 billion bioactivities with spike exclusively and other main proteins of SARS-CoV-2 like main protease, okay, the RNA-dependent RNA polymerase. So these are other important proteins, protein targets of SARS-CoV-2. We have exclusive kind of literatures for each and every proteins also. We have already developed COVID-19 docking server developed in April 2019 itself. Like if you have any plant compounds, you can check with any of these receptors by simply uploading the chemical compound in SDF or SDF format. Okay. So SDF format, if you upload and if you put your mail, obviously you receive the results also. So all these things, okay. So this kind of a COVID-19 docking server, of course, developed using the machine learning technique. And this is a click-through drug pipeline and a pipeline of artificial intelligence based drug discovery development of COVID-19. So according to Akanksha et al., okay, she has used a kind of a deep learning model, okay, and she linked with virtual screening strategies to discover a drug for the targets like spike glycoprotein. Even they have targeted the human ACE2 main protease, RTRP, as I already mentioned. Okay, in protease, we have chymotrypsin like protease, papain like protease. So types of proteases are there. Okay, chymotrypsin and papain like protease. So these are the important proteases considered as important targets for SARS-CoV-2. Okay. And computational drug repurposing model. So also she has developed a kind of network-based algorithms, and based on the networks, okay, the CoV to associated host proteins and other proteins. So she also developed a kind of a network models, and also based on the expression, gene expression, okay, with the normal as well as SARS-CoV-2 infection. Okay, so see here in SARS-CoV-2, you find more blue dots, isn't it? So from this, you can able to find the infection level is more in SARS-CoV-2 when compared to the normal as well as the other kind of a control, ACE2 inhibitor. Okay, and if you see, she also has used the homology modeling, which is other kind of a drug design methodologies in her pipeline to discover the drug for SARS-CoV-2. So this is one more pipeline, okay, where we start with a drug, and we if you want to discover a drug, okay, generally for personalized medicine, okay, we have to check with different kind of a patients, isn't it? Some patients are symptomatic, some patients are asymptomatic, and some find to be pure healthy. If you want to check, okay, with the clinical trials, okay, from phase 1 to phase 4, obviously the drug effects varies with respect to individuals. So this is also an important criteria to be discussed. So we can use this kind of an integrative context to find a drug, okay, for SARS-CoV-2. And in this slide, you can able to find a drug to be discovered for a personalized medicine. Okay, like if you want to discover a personalized medicine for SARS-CoV-2, obviously we have to take into account of genomic analysis, transcriptome, proteome, phenome. All these 4 analyses are important. For example, if severe COVID-19 patient is indicated in green color, according to this, we have resistant to infection in pink. The asymptomatic we have in blue color and symptomatic in orange color. So to distinguish exactly, okay, so we have to take all this kind of a profiling, and from the profiling patterns, from the we can able to identify, can provide drugs according to that, because whatever drugs we provide for symptomatic, okay, we can't or symptomatic or asymptomatic, we can't able to give it for severe COVID-19. We have to give the corresponding drug. Okay, first we have to identify the nature of infection. Based on nature of infection, we have to provide the drug. So for this, this kind of an artificial intelligence helps you. So how the artificial intelligence helps, like as I already explained you, take the X-ray from the patients, and from the lung infection, we can able to segregate the data. We can categorize the data, and from the data we can prescribe a kind of a drug. And one such event happened in 2020. So where Dr. Girinath Pillai is one of the main mentor for 12,000 teams participated in this kind of an event. In the 12,000 teams, so only 53 teams, okay, got selected for internships. So one of my team, okay, so from third years and second years, okay, though they worked essentially on this problem statement, that is on identifying covalent spike protein inhibitors to control the infection of SARS-CoV-2 from small molecule databases. So they just worked on the both closed state spike glycoprotein and open spike glycoprotein and came up with a kind of a drug. Now, I think yesterday they received a mail around 3.6 lakhs generated, okay, as a product of internship. Okay, they'll be getting as an internship while acquiring a training from the drug discovery hackathon team. And you know what is the methodology we've used? We have just taken 1,000. This is a very simple methodology we have used. Okay, a simple cracking technique. What we taken is we have taken a plant compounds and natural compounds from NCBI. At that time we had only 1,152 compounds. Now we found 1,600 something, isn't it? At the time we had only 1,152 compounds. We downloaded as a data set, and then we find a Lipinskaya rule and Swiss ADME, and so we've filtered around 400 compounds, and from 400 compounds we have taken 100 best compounds from doing virtual screening through Autodoc Pyrex. Okay, so how we have done by docking the kind of a plant compound, the compounds what are downloaded from MCP PubChem data set with closed spike glycoprotein and open state glycoprotein. So from this, we identified only 20 base compounds. We identified the best 20 base compounds based on the internal energy. So these 20 base compounds we used further for manual docking to find the hydrogen bond interactions as well as kind of a binding site amino acid. So from that, okay, we docked complexes, we passed it to the simulation, the top 3 compounds we passed to the simulation, where we found out the best compound as Vithaferin A. Okay, so this is a plant compound obtained from ashwagandha. We call it as Withania somnifera. Okay, Indian ginseng. So this is the plant compound. This is the product we got, which is an outcome of drug discovery hackathon event. You know, it's already cleared to phase 2 trials, and even IIT Delhi Professor Sundar was explaining that this plant will be a cure. Okay, so he is suggesting this plant in earlier 2019. Okay, so for past 2 years, he has done this kind of researches, and is still claiming that this plant compound can be a cure. Okay, like a remdesivir. And this is a kind of a molecular docking interaction patterns, and this is a simulation graphs we got from the simulation using Chromax. And this is a kind of a machine learning technique. Okay, we can develop using Python programming or R programming. If you see in R programming, okay, if you want to go for chemometric calculations, like in cheminformatics, kind of a softwares can be developed, okay, by R programming or Python programming. So here I have shown you a list of packages: RCDK, chemometrics, RJava, Chemine. These are some of the packages, okay, present in R. You know, around 15,000 packages are there in R, okay, for doing the chemical cheminformatics studies, and even we have some modules for docking. Especially we have more for statistics. We have more statistical tools in R program. Okay, so the binding analysis and the property chemical parameter analysis, descriptor analysis, all these things can be done. Here I have just shown an example for cheminformatics. That is, this is a chemistry R programming chemistry development kit. Okay, chemometrics determines you the various parameters like log partition coefficient, molecular weight, whatever you see for a compound. All these things can be determined from this. So if you want to use any of the functions, we can use this package. So to import a package, like how we import a header file in C programming, we can import this kind of a package using library function. So we find if you want to work with this kind of 2D or 3D structured chemical compounds, we of course need a kind of a Java platform. So we are also importing the R Java. Okay, initially you install the packages, and if you import using the library function, then you can utilize all the functions present in this package. Okay, so, and one such function is also mentioned here. It's a load dot molecule. So if you want to load a series of compounds present in a data set, you can also write a code like this. And if you want to view molecule in 2D, you can write like this, okay, where you can, if you want to put in the form of a table, okay, so all these parameters are mentioned according to that. Likewise, not only that, if you want to screen a compounds based on a particular molecular weight, okay, molecular weight less than 500, and the log partition coefficient less than 5, something like that, even you can do it, okay, using this kind of an R program. Whatever you do in the software, okay, for developed using R programming, and in Python, majorly used for image acquisition, okay, the analysis. And similarly, we have developed a kind of an immunoanalytical database in 2017, okay, where this is a database for developed from plant-based adjuvants. So whatever database we have developed, okay, we can use it for used to discover drug for any type of cancer. Okay, so we generally have to feed the kind of a molecules and we import the kind of a data. And if you try to analyze it, obviously you can find, for example, if some cytokines, for example, IL-2 or IL-4 is responsible for asthma, if you select a kind of a protein, so obviously you can find the list of compounds which is majorly interacting with kind of a plant compounds. Okay, something like that. Okay, we developed a plant-based adjuvant database interacting with immune proteins. Especially we used cytokines. More than 1,000 interactions we have uploaded it, and the kind of a docking information obtained from Discovery Studio Ligand Fit, we use it as a data to upload in SQL Server, and we have used a front end as C#, okay, to project the data in the screen. Okay, so these are a kind of an online kind of a thing. Soon we'll be posting in the website. Okay. And the summary, nearly 30% of the marketing drugs are repositioning drugs, and spike protein plays an important role in viral entry by interacting with the AC2 receptor of host cell. So the transition to the semi-open state is a critical step in host cell recognition. We can use artificial intelligence algorithms to find how a drug can interact exactly in the semi-open state or in the other states. And Vitafrin is a compound we screened in the drug discovery hackathon event. Okay, so it almost found to be binding 4 out of 13 potential binding sites, also showed a significant orthodox score, and also it clearing up to the phase 2 clinical trials. And these are some of the events. Almost I've completed my presentation. I will take into the hands-on. Before that, I'm just showing you like these events are conducted in this year. Okay, so genomic data science and precision medicine completed. This is a 15 days online course. And in the next slide, okay. Okay, so the 15 days course I delivered in other institutes, and the 5 days course later to the drug and vaccine designs I have delivered. There is a recent webinar delivered in an industry. And we are going to organize an upcoming guest lecture in anti-cancer drug discovery. We got DPT-CTP sponsored for popular lectures. So Girinath Pillai is one of the speaker, and also we have CDRA, Dr. Sanjay Batra, and Professor MC1. So now I think I thank to the management. Before going to express the thanks, I can go ahead with the online. Can I go for 15 minutes, ma'am, with hands-on experience?
Yes, sir.
10 minutes?
Yes, sir.
Okay,
Yes,
thank
sir.
you. So I think I am share the screen. Okay, I'll be taking you to the Autodoc Parex. So whatever we have used in the drug discovery hackathon.
Excuse me, sir.
Yes, ma'am.
Sir,
I have to share it.
the screen is, yeah.
Yes, ma'am. I'm coming. So I'm sharing my screen. This is my screen. Yes. Oh, fine. Yes. Now I found the screen. Is it visible, ma'am? My screen?
Yes, sir. It is visible.
Okay. So now I'm going to take you to how to discover drugs which using visual screening technique. So initially I go with PubChem. Example I'm showing. So initially I want to find a COVID-19 data set, or if you know a particular compound, okay, for example, Vitafrin A, okay, this is the result of my research. So I'm just taking the Vitafrin A, okay, so along with the Vitafrin A, I also find 27 related compounds with respect to Vitafrin. Okay, so I don't want to take all the 28 compounds. I want to take a few compounds based on a molecular weight. For example, if you want to screen compounds based on molecular weight, like for example, 450 to 470. Okay, so from 28 compounds, I got 22 results. Okay, whatever results I got, okay, so I'll be taking this. Okay, I'll be taking this and I'll be downloading it as a data set. So based on a specific parameter, whatever you fix it, and you can download that list of compounds in a single SDF format. Okay, so I'm not using a 3D, I'm simply using a 2D. Okay, in a desktop, I'm going to create a folder called demo 7. Okay, in a demo 7, I am going to create a kind of a ligand kind of uh, a file. Okay, ligand a data set. In this, I found two compounds. What compounds? Dihydroxyvitaferin and vitaferin A. Okay, so two compounds I have. I'll be checking which compound is best for my receptor. So already used open state glycoprotein. Okay, so the open state glycoprotein, I think I have downloaded somewhere here. Okay, go to the PDB website and download the protein files that I have used 6V by B. Okay, 6V by B. I have plenty of chains. I'm going to take just the A chain of my spike glycoprotein. Okay, and if you are working with alpha variant, just mutate the sequence. Mutate the sequence, and then you go for homology modeling using Swiss modeler. And after modeling the protein file, okay, you take that as a target receptor. Now I explain you how to do the virtual screening using Autodoc Parex. Okay, so Autodoc Parex. You simply have to initially load the molecule. That is the first step. Okay, so before that, if you want to set the parameters, okay, to the default get an output in a particular folder, you feed it. So presently I have a folder called demo 7 in desktop. So in workspace, I clicked edit and Autodoc preferences. You got this kind of a tab. So in desktop, I'm going to feed my I've fed my folder, so I'm giving the correct path. After feeding the correct path, click OK. Okay, I fed the path now. So whatever output you're going to generate, it will be generated in the demo 7 folder present in desktop. Now the next step is to load the molecule. So click File and Load Molecule. So after clicking Load Molecule, okay, go to the corresponding desktop. Demo 7 is my folder, isn't it? Where I have both input file, ligand and receptor. Initially, they are asking you to load macromolecule. So what is the macromolecule? Protein is our macromolecule. Obviously, ligand is the small molecule, isn't it? So if you want to load the macromolecule, open the protein.pdb. So this is a open state spike glycoprotein. Okay, so I just uploaded a kind of an open state spike glycoprotein. Now you have the next step is to load the ligand. So to load the ligand, go to the open babel. Inside the open babel, you have an option like this. You click here, okay, and inside this, go to the desktop. Okay, in desktop you have demo 7. Go to that demo 7 where we find ligand.htf. You remember we have two different with vitaferin A derivatives. Okay, in stored in a single STF format. So in open babel, I'm clicking here, and you are getting this kind of an icon. I'm simply opening it. So this is the ligand I got. Okay. So we also have a second ligand also. So the next step is to click here and right click here. You get something like minimize selector. Okay, this option we use for doing energy minimization. If you want to do energy minimization for this compound alone, you have to select this. But if you want to do energy minimization for all the compounds, for example, here I just downloaded only 2 compounds. Suppose if you are downloading all the 28 compounds related to vitaferin A, simply click minimize all the 28 compounds will be done energy minimization soon. Just because I want to complete soon, I'm just taken 2 plant compounds. Now I'm clicking minimize all. So while doing the energy minimization, you see the variation. So happened with respect to the compound. I think I have done for the first compound. Now energy minimization for the second compound has done, isn't it? So the energy. So now you got an energy minimized ligand compounds. Now the next step is to go for the uploading the ligand. Okay, so to convert all ligands to Autodoc ligand PDB. So before that, you can also convert this. Okay, you can make this receptor as macromolecule, because initially we had loaded the protein receptor, isn't it? So there is one more step left out, that is make this molecule as macromolecule. Oh, sorry, it happened wrongly. Oh. I think we got some problem. Can I go for expanding once again? So I can take you. I think it takes just 5 minutes. Just 5 minutes, I can complete my hands-on. So again, I'm telling we already had a kind of an imported kind of a preferences. We have already imported a path, so no need to do this. So first step is to load the molecule. So go to the desktop, go to the demo 7. In the demo 7, we go for the protein.pdb. Okay, if you have uploaded the protein.pdb, and what we have done, we have to click here. We have to make this molecule as macromolecule.
Screen.
Oh, screen is not visible, isn't it?
Yeah.
Okay, I share the screen. Share screen. Okay, this is my Autodoc. Yes, I just completed making my protein as macromolecule. The next one is to load a ligand. Okay, so to load a ligand, I'm going to the desktop, I'm going to the demo 7 folder. In demo 7 folder, you open the ligand.stf. I got 2 ligands, and you simply click here if you want to do energy minimization with these 2 plant compounds. Okay, so click minimize all. Now energy minimization is going to done. Okay, some kind of conformational changes we can observe. Okay, after this, okay, you have to click here a ligand, and you can go for conversion of Autodoc ligand in the form of PDBQT. You know, the Autodoc uses PDBQT as formats to doc, okay, both ligand and receptor. So we have converted that. So after converting both the ligands as PDBQT, go to the Vina wizard. Okay, so after going to the Vina wizard, you have to select the ligands. Okay, we have 2 ligands, and in protein we have protein.pdbqt. We have selected all the molecules now. Now click start. Okay, and again you can select if it is not selected properly. Yes, I have selected. Click forward. See, you will be getting a kind of a grid in the protein receptor, where if you click forward, obviously a kind of a docking is happening on. Okay, virtual screening is happening on with the first ligand. Okay, now our vitafrin A derivative is docking with one of the open state glycoprotein. After this docking, okay, you can find the docking of second ligand. Okay, you'll be getting a series of energy results after the docking. Okay, so if you see the kind of a docking results, earlier I did the same. Okay, if you download the internal energy values, you can find like this. Okay, so generally the first pose share whether you can able to see the Excel sheet. See the Excel sheet. So this is the output of the virtual screening using Autodoc Parex. So you can find a ligand is binding with the receptor. The first ligand is the best pose is -7.3, and the second ligand is -6.9. This is how we can screen it. Of course, this is done okay using a combination of machine learning techniques. Okay, of course, I've completed my presentation. So any queries you can ask me. So these are some of the grants received. Did I have to share this also? Yes, I've shared.
Delegates, you can post the queries in the chat box, or you can unmute yourself.
If you have any questions, you can ask me related to drug design, repurposing, artificial intelligence. I think I have linked almost all the 3 in a single 1.5 hour session.
Exactly, sir. Exactly, and very lucid presentation. Actually, you know, you started with the basics and then executed well with some hands-on on Autodoc Pyrex, which is most user-friendly, and people are very much using nowadays actually Pyrex for virtual screenings and all, and touched to some of the toxicity softwares also. Very wonderfully you taken away the topic actually.
Sorry, but actually I'm not proficient with the Zoom platform. So on each and every time I have to share that only I'm learning now. Okay, but any doubts if you have, you can ask me, or if you want to show me some slides, I can show you. Okay.
Delegates, if you have any queries, please post in the chat box.
So I'll be passing this to your WhatsApp group with your permission, ma'am. So like
Sure, sir.
the participants willing to participate in this popular lectures, they can attend this lecture.
Sure, sir. Sure, sir. And sir, there is a one question. Can we use Python for docking?
Yes, the Python is the best tool for docking when compared to R. R is majorly meant for statistic analysis. In Python, for docking, we have specific modules. The Python is linked with almost all the languages. If you want to link with the internet, okay, we can link with the Java platforms. Okay, and if you want to do the statistics, we can link with also an R. So there's a more this is a versatile software, okay, can portable software link with all operating systems. We have plenty of modules in Python. So docking is what actually? The docking is checking the binding affinity through calculation of internal energy, Gibbs free energy. Del G equals del H minus T del S. So it's a thermodynamic calculation. So using a thermodynamic calculation and finding the structural kind of an affinity, that is structural oriented thing, we will be deploying a kind of an algorithm in Python. Okay, so we have specific modules to do molecular docking using Python. Discovery Studio is developed through Perl programming. In Discovery Studio, some docking and finding through the blast is done through Perl. But when compared to Perl, we have so many modules, integrated modules are developed using IDE or developed through Python. So it's a base software. BioVia, Scroid Engine or MOE, which is best? Actually, BioVia, we have very less number of steps for doing docking, QSAR, simulation. We have very, very less number of steps. For docking, we have to take only 5 to 7 steps. If you want to do extensively for calculating hydrogen bond interactions, finding the good quality images, okay, we may go for 7 steps. We don't have even minor steps, okay, only 7 major steps you have, that's it. But whereas if you go for Autodoc, you have so many minor sub-steps, okay, so each and every sub-step you have to do manually. So it's an automated BioVia. And Scroid-Incher, if you have 7 steps for docking in Scroid-Incher, you have to do 9 to 10 steps. Okay, but Scroid-Incher, the quantum mechanics derived from the Scroid-Incher wave equation, isn't it? So we have wave equation. The quantum mechanics rely on Scroid-Incher wave equations. So of course, the Scroid-Incher is the software, the first software developed using with this kind of a quantum mechanics. We have some few additional steps with the Scroid-Incher. Okay, we have few parameters which we can able to find in Scroid-Incher, which BioVia found, and also BioVia have few parameters. For example, docking, you can generate 8 different scores. In Autodoc, we can get only 1 score, internal energy score. But when Scroid, in BioVia, we can get 8 different scores using BioVia Discovery Ligand Fit. If you want to do multiple ligands with a single receptor, we also have a module in BioVia. We call that as LibDoc. LibDoc, we use multiple screening of ligands with a single receptor. Okay, so all this finding can be done within a few minutes. Okay, even in Autodoc, I showed you a result, or I don't know. It will be getting a result within a minute. Okay, the 2 components talking with result. But the same, if you do manually with an Autodoc, it may take a minimum of 40 minutes to 1 hour. Okay, we have the virtual screening softwares are rapid resulting performance. Okay, it's a fast acting performance. So initially you can do with virtual screening, then you can go with a manual to find hydrogen bond interactions or kind of binding site predictions. And what is MOE? Can you mention what is MOE? DFT in drug design. Can you something in DFT? It's a Fourier transform, isn't it? So in for Fourier transform, yes, of course, this role is needed. Or you have any specific expansion with respect to DFT? Can you unmute and answer, or you can chat? molecular operating environment. Okay, molecular operating environment. It's not a popular one. We have BioVia Discovery Studio, BioVia, Scroid Ninja, Triposibal. This is also a popular kind of a software. Majorly, these softwares, they use it. Yasera, and these softwares are not that much popular. MOE, molecular operating environment, is of course okay. But this kind of a molecular operations can be controlled using Python modules. Okay, can integrate with the Python modules. It can work. And anything about EFT? In Python, like if you are taking care of calculating lipophilic parameters, toxicity, if you're not accounting for toxicity, even if you want to calculate toxicity based on a combination of parameters in combination of chemical properties in consideration, okay, you can develop your own docking software. The docking software you can develop by linking with whatever modules you want. Was Euler very famous before? Okay. But in Python, they are extending the features, more features. They are improving the docking algorithms, because now we can't rely on the in silico approaches. But while you do experiment with animals, by getting an institutional animal ethical committee permission, they are telling initially you have to do all the experiments with the computational. So if we rely on computational, we have to think about how much precise our softwares work based on the docking algorithm they've implemented and the scoring function they've deployed. So we can improve our software. Okay, we can by using the improved algorithms, or we can use a pipeline. Like the Dr. Girinath Pillai is explaining a view about how to convert one format to another format, which is happening with Open Babel software or Babel Bin. Okay, here he also explained how we can create our own kind of a modules linked in neural networks to convert the formats. So that's a simple example. Like this, even you can develop your own docking software by fixing an algorithm and the mission and the technique. Okay, even if you go for binding site prediction, we can use flood fill algorithm, or we can use other kind of a random forest kind of an algorithm. So use an algorithm, select an algorithm, and select a scoring function, select a kind of a descriptors for screening. Then you can go for docking. Everything is now it's something like it's in your hands how to do a docking. Okay, so this can be happen nowadays with Python and R programming. And any other doubts?
Yeah, we have another one more question.
Density functional theory. Okay, actually, this is one such theory, density functional theory. Like, however, we find different machine learning algorithms, isn't it? We find different. Even if you see the docking, okay, in docking we have earlier lock and key, induced fit, conformational theory. Okay, so likewise we have we come across up to the conformational ensemble method. Okay, so of course this density functional theory is of course one of the method. We can use this as one of the method for drug design. So if you want to screen multiple compounds through high throughput screening, okay, high content screening, we of course need this kind. What about V-Life MDS? V-Life MDS. So MDS, but actually I'm hearing this software something new, but very popular eminent softwares, or what the ChemSketch, ChemAxon, Marvin ChemAxon, you remember? The R programming module, what I shown in the slide, is a product of Marvin ChemSketch, ChemAxon. Yesterday you heard a lecture from an industry person, isn't it? So he explained whatever he explained, and whatever a software developed by him is from R programming. Okay, so I think I have to check about V-Life MDS. Presently, I don't know about this.
So another question from Vinay Kumar is for repurposing non-clinical or in silico studies required.
I'm not getting, ma'am. For clinical, we have
non-clinical or in silico studies required to conduct again for repurposing.
Non-clinical studies. Actually, whatever we try to do, the repurposing is non-clinical. But till now, I didn't explain you something about clinical databases and utilization of clinical data for screening the drugs. We have exclusive kind of a clinical databases. From clinical databases, we can get the expression data, how much they have cured, etc. Okay, so from the expression analysis, okay, we can take the data, we can select the target receptors, okay, and we can go for that. Okay, but non-clinical is what already there as they just that is a target based. Okay, it's not exactly a clinical the therapy based. Okay, so target based is what I was explaining from the beginning. The therapy based, you have to look through the clinical data. Simulation+.com. I'm not exactly. Oh, VLife MDS is molecular dynamic simulation. VLife, very good. Yeah, actually, recently in most of the publications where you find more flexible kind of a data with NAMD. That is the most preferred kind of MD simulation software. But for purchasing the simulation server, the cost around comes around 6 lakhs. Okay, so we have to spend for 6 lakhs for the server with a graphics card. Okay, but if you go with a Promax, obviously it's a freeware, so it can reduce the software price. But if you go for such a models like NAMD, I don't know about VLife MDS. I have to check whether it's a commercial or not. Then you have to invest for something for that also. But we can get excellent results even from the freeware like Romax. And also, Dr. Girnath Pillai has a kind of a simulation in his repository. I think whatever simulation we do, it will be there for 12 hours. Okay, after the data will be automatically deleted. So I suggest you to rely on purchasing a server, installing a software like a Promax, a freeware, and you can do with a kind of a simulations. Any other doubts?
I guess no, sir. And I couldn't find any more questions in the chat box. Thank you for your patient explanation to all the inquiries raised by participants.
Thank you, participants. I didn't. You have interacted very much. So I am pleasant to find more questions from your side, and even I am learning from you. Like these softwares are available presently to do all this kind of a drug repurposing, docking studies, etc. So I'm happy that you have interacted well. And if you have any more questions, you can raise it to my email ID. Okay, so I have provided my email ID. So you can reach to me. Actually, I've registered as a PhD supervisor. Okay, so even if you want any kind of a support with respect to guiding your students, okay, so I can provide you a support. Okay, we have a Discovery Studio commercial license in our institution, and I thank SVC management and Seven Hills College of Pharmacy and especially AICTE to sponsor the ATAL-FDP on this theme. So thank you very much. If you don't have questions, you can raise it to my email. Raise your queries.
Sir, kindly stop sharing, sir.
Yes, ma'am. I think I stopped sharing, isn't it?
Yes, yes, yes,
yes.
Thank you. And at this juncture, hello, sir.
Hi, ma'am. Your voice is not audible. Hello. From my side, you have internet disconnecting?
Yeah.
Okay. No, only from you.
Can you hear me now?
Yes,
can you hear me now?
Yes, yes. Now it's audible to me. I can find internet frequency fine.
Yeah. Thank you, sir. Thank you so much, sir, for the wonderful talk. And at this note, I want to thank Divya Madam also, because she's the one who just shown me a way to reach out to you. And thank you so much for addressing all our delegates today with your wonderful talk. And From the management and principal sir, all the people and staff members, I just take this opportunity of honoring you with a certificate and memento, sir. Kindly accept it virtually, and the same will be posted
Thank you,
to
ma'am.
your mailing address.
Also suggest all the participants to utilize these kind of utilities. We can get plenty of funds. We can generate plenty of funds with it. So my mightiest support will be there for any kind of a project's guidance. So thank you, Madam Sucharita Madam and Dr. Divya Madam, who has provided me an opportunity. Also, I thank your college management and my college management at last. Have a great day.
Thank you, sir. Thank you so much, and much inspired from you as a eminent speaker, and also you have a keen interest in listening from all other eminent speakers too in this ATAL-FDP program. So inspiring to have you on this platform, sir. Thank you so much, and bye-bye. Dear delegates, it is up for this today, day 2, and tomorrow we can join here in the same platform of Zoom with the same link exactly by 10 a.m. And as usual, tomorrow we have 3 sessions, morning 2 sessions and afternoon 1 session. And I think Praveen sir, I think sir is there, and sir, there is a one question.
I provide you information in WhatsApp web.
Yeah. Yeah, how to register for upcoming event in November. So kindly share those details in the WhatsApp group, sir, so that people can get benefit out of it.
Also, we'll be conducting the online certificate courses. I provide this information WhatsApp web and group email IDs. So each and every month we are conducting 2 or 3 kind of an events related to drug design and vaccine design. Already I've guided a project in Python, kind of a tensor workflow. So similar kind of a certification programs in Python or programming will be, you can expect these kind of programs from our institution. So thank you, ma'am, for providing me this opportunity to share the information.
Thank you, sir. Thank you so much. And dear delegates, that's all for today. And tomorrow we can join here for 10 a.m. Thank you. And if you have any queries, you just write it to me, or you can unmute now and you can just tell it out. The feedback film will be posted in the WhatsApp group after completion of this session. Any other suggestions or any other queries for the delegates? I hope no. And to this, I thank once again Praveen sir for joining, and see you tomorrow, sir. Bye.